Hi, my name is Gray Redding, and I'm a master's student here at the Chesapeake Biological Lab. I work with Dr. David Secor. I've been here for about a year now, and I'm working with Atlantic mackerel, trying to use stable isotope information that's held in their otoliths to figure out how they're moving around and how all the different groups of this fish might be mixing together. So here at CBL, I study Atlantic mackerel population structure. So Atlantic mackerel are a fish that's pretty common in the more northern parts of the Atlantic, all the way from near America to over in Europe. And with this fish, there's a lot of questions about how it's sort of organized across that space. Sort of if there's different populations or different groups that are living in different areas of that space. And so I'm trying to use stable isotopes in their otoliths, which are basically these sort of chemicals that we can use to identify sort of bodies of water that the fish might have been in. I'm trying to use that information to figure out what group individual fish might have come from. So with my research, I hope to be able to make recommendations for management. If we could have a better idea of where these fish are going, where they're from, and where they might end up, we could get a better better numbers and guesses for stock assessments that we do. And these stock assessments help us set the levels of fishing that we think are sustainable. The levels of fish we might harvest through commercial fishing or recreational fishing, or sort of how hard we think we should fish the population, or sort of if we should set certain dates where we should or shouldn't fish. And so with my thesis, I hope to be able to add to that information and sort of create more sustainable management for this mackerel population.